In the name of God, in this session I'll talk about the complement system. You will learn how the complement system works in our body and the proteins in the what are the proteins in the complement system, and what is the role of complement system in innate and acquired immunity. And there are proteins in the complement system which regulate the activation of complement system. I'll talk about the regulatory proteins in the complement system and how these proteins regulate the complement system. And finally, I'll talk about the consequences of deficiency of complement proteins. I want to start my lesson with a case. This is a case, a 25 years old patient who admitted to a hospital with pneumonia and Haemophilus influenza was isolated from his sputum. And in his history, he had repeated middle ear infections, and the different bacteria, pyogenic bacteria, were isolated from his ears, and including Staphylococcus aureus and Proteus vulgaris and Pseudomonas aeruginosa. These are bacteria which isolated from his middle ear. And he had two septicemia due to hemolytic streptococci and also due to Neisseria meningitis. The serum, his serum was analyzed for antibodies and complement proteins. As you see here, all the antibodies levels were normal. IgG, IgA, and IgM levels, serum levels were normal in this patient, and also this patient responded normally to tetanus toxoid vaccine. And this shows that the humoral immunity or B cells are normal in this patient. But the serum levels of complement proteins such as C3 were abnormal. For example, here you see that C3 were 27 milligram per deciliter, but the normal range of C3 in our serum is 97 to 204 milligram per deciliter. But other serum levels of other complement components were normal, but factor B also were abnormal, and the level factor B was undetectable in the serum of the patients. All of these results shows that the complement, there are a deficiency in the complement proteins in this patient. And this deficiency of the complement proteins is the cause of the repeated or recurrent infection in this patient. What is the complement system? As you see, the deficiency of the complement system can cause repeated infections, and how the complement system prevent the repeated infections in our body. Now, complement system consists of a complex series of over 30 soluble and cell-bound proteins, and many of which are enzymes, but enzymes are inactive in our serum, and the inactive enzymes are called proenzyme or zymogen. And the proteins of these enzymes in the complement system has the proteinase properties. They are proteinases. And the biological activities of the complement system affect both innate and acquired immunity. Then the complement system is very important in both innate and acquired immunity. Some proteins in the complement system can activate after binding to an antibody molecule, but some activation pathways in the complement system are independent to the antibody for activation. I'll talk about these matters in the later slides. Complement components are produced by different cells in our body. For example, the main producer of complement components is hepatocytes and macrophages, monocytes in the blood, and epithelial cells of gastrointestinal and other mucous membrane can produce complement proteins or components. In this slide, I want to notice three points that are important for explaining of activation pathways. One of them is the nomenclature of the complement proteins. As you see here, complement components are designated by numerals, letter symbols, and trivial names. As you see here, 9 proteins in the complement system named by numbers C1, C2, C3, C9 till C9. And some of the complement components named by letters, for example, factor D, factor B, and some of them named by trivial names, for example, homologous redirection factor. This is a regulatory protein in complement system, and it is named by a trivial name, homologous redirection factor. Another point is the fragmentation of the complement proteins. As you see here, C4 is a component of complement system, and it is cleaved by C1s. C1s is an enzyme in the complement system and cleaves C4 into C4b and C4a. The smaller fragment is designated by a small a, and the bigger fragment is designated by a small b. Then the bigger fragment is shown by a small b, and the small fragment is shown by a small a. And but there is an exception here for C2. For C2, the bigger fragment is shown by C2a, and the small fragment is shown by C2b. And after activation of the complement system, some fragments bind together and form a complex, and this complex has a enzymatic property. For example, here C4B2A is composed of two fragments, C4B and C2A. And this complex has a enzymatic property, and this enzymatic property is shown by a bar over the number and symbol in this complex. Complement activation, complement system activates in three pathways, via three pathways: classical pathways, MB-lectin pathway, and alternative pathway. As you see here, three pathways has their own proteins, and some proteins are common between three pathways. For example, C3 protein is common between three pathways, and C4 and C2 are common between classical and lectin pathway. As you see here, the three pathways, classical pathway, lectin pathway, and alternative pathway, are different in a part of activation, and they are the same in the final part of the activation. Then we can divide these three pathways into two phases, the early phase and the final phase. And as you see here, the early phase of the three pathways are different, and they produce different enzymes or complexes which have enzymatic property. For example, here C3 convertase produced by classical pathway and lectin pathway, and it is different with this C3 convertase which produced by alternative pathway. And also these three pathways produce another enzyme called C5 convertase, and this C5 convertase is different between alternative pathway and lectin pathway and classical pathway. Then in this lesson, I'll explain classical pathway, early phase and final phase of classical pathway first, and then I'll explain early phase of lectin pathway and alternative pathway. Okay, classical pathway. As you see here, in the activation of classical pathway needs antibodies. Just classical pathway needs antibody for activation, and from antibodies just IgM and IgG classes can activate classical pathway, and in between IgG subclasses IgG1, IgG2 and IgG3 can activate classical pathway. The first protein initiate the activation of a classical pathway is C1. C1 just work in the classical pathway. As you see here, C1 composed of three. C1 is composed of three subunits: C1q, C1r, C1s. Of course, C1r and C1s have two subunits. Then there are two subunits of C1R and two subunits of C1S and one subunit of C1Q. As you see here, C1Q has a collagen-like structure with a stalk and 6 heads here, and it binds to antibody. C1 binds to antibodies from the head of C1Q, heads of C1Qs. As you see here, these are IgGs. These are molecules of IgG, and the IgG, when binds to a antigen, form a antigen antibody complex. After forming of antigen antibody complex, the C1 can bind to the Fc of the antibodies, Fc region of the antibodies from its head, C1Q head. Then C1 binds to the CH2 region of the antibodies IgG from the head of C1Q. For activation of classical pathway, two head of C1Q must be bind to the Fc region of two molecules of IgG, as you see here. For IgM, because there are two CH region in one molecule antibody, because IgM is pentameric, and we have there are 5 Fc region in one molecule. Then C1 can easily bind to 2 CH or Fc portion of the IgM. IgM in the serum exists in this feature, planar feature, and when IgM binds to the antigen, the Fc region exposed, and C1 can bind to the Fc region of the IgM and activates, and complement system activates. Okay. After binding of C1Q to Fc region of the antibodies on the surface of the antigen, the C1S activates, and C1S can C1R, first C1R activates, and C1R after activation can activate C1S, and C1S after activation has two substrates, C4 and C2. First, C1S cleaves C4 into C4b and C4b and C4a. C4a diffuse away, and C4b binds to on the surface of target cell. After then, C2 binds to C4b, and again C1S cleaves C2 into C2a and into C2a and C2b. Which of the is the bigger fragment, and which of them is the smaller fragment? Yes, the C2b is the smaller fragment and C2a is the bigger fragment. Then the bigger fragment binds to C4b and forms a complex, complex C4B2A, and this complex has enzymatic property. That's why the enzymatic property is shown by a bar over the 4B2A. And this complex is called C3 convertase. What is the function of C3 convertase? Yes, C3 convertase can cleave C3 into C3b and C3a, and C3b bind to C4b2a and forms another enzyme called C5 convertase. Then C4b2a3b is the C3 convertase of classical pathway, and C2a the smaller fragment released in the environment. Okay, you can see classical pathway at a glance in this slide. And here, after forming of C5 convertase, is the end of the classical pathway, the end of early phase of classical pathway. And here you can see the starting of final step or final phase. Then cleavage of C5 into C5b and C5a is the start of final step. Then here C5 convertase cleaves C5 into C5b and C5a. Again, C5b binds to the on the surface of target cells, and C5a release and diffuse away. Okay, after forming of C5b on the surface of the antigen here, another complement components until C9 binds to each other or interact to each other sequentially. As you see here, C6 binds to C5b. C6 can stabilize C5b, binding of C5b on the surface of the antigen or target cell. And after binding of C6, the C7 binds to C5b6, and C7 after binding undergoes conformational changes, and the hydrophobic moieties exposed on the surface of the molecule, and after exposing of hydrophobic sites, the C7 can insert to bilayer of membrane of the cell membrane, as you see here, and the hydrophobic sites of the C7 bind to the hydrophobic site of the bilayer of the cell membrane. And after then C8 again bind to C5b67, and again here C8 also undergoes conformational changes, conformational structure, and the hydrophobic moieties exposed on the surface, and again here C8 can insert the bilayer cell membrane more, and after then 10 to 17 molecules of C9 polymerize around the C5b678 and form a big hole in the bilayer of the cell membrane. And this hole is called membrane attack complex. And this pathway from cleavage of C5 into C5a and C5b until the formation of MAC is called MAC pathway or membrane attack complex pathway. Okay, this is an electronic microscopy picture. And you can see here a big hole on the surface of target cell, and it is taken by electronic microscope. And in this slide you can see the membrane attack complex pathway at a glance. And another pathway in the activation of complement system is alternative pathway, and the initiator of the alternative pathway is C3b. C3b is formed by the hydrolysis, spontaneous hydrolysis of C3 in our body, and this C3b that forms from the hydrolysis of C3b is shown in this way, C3H2O. After entrance of the microbe in our body or any foreign substances in our body, C3b binds to the surfaces of the foreign substances, and the alternative pathway initiates. After binding of C3b on the surface of the, for example, a bacterium, the factor B, protein in the alternative pathway, specific for alternative pathway, binds to C3b, and enzyme called factor D that is specific for alternative pathway also cleaves factor B into B and BA. BA diffuse away and BB binds to C3b and make C3 convertase of alternative pathway, and this complex is C3 convertase of alternative pathway. And this C3 convertase also can cleave C3 into C3a and C3b. C3b binds to C3bbb and form C5 convertase of alternative pathway. Then C5 convertase of alternative pathway look like a sandwich. In two sides there are C3b and in the middle of the molecule BB, and this is C5 convertase of alternative pathway, and this molecule has enzymatic property, and it is shown by this bar. Okay, you can see in this table different surfaces or different microorganisms that can activate alternative pathway. As you see here, different gram-positive and gram-negative bacteria, and also fungal, yeast cell walls, and viruses, and tumor cells, parasites. All of these can activate alternative pathway. And one of the best activator of alternative pathway is a cobra venom factor that can activate alternative pathway seriously or severely. The last pathway is lectin pathway. Lectin pathway is similar to classic pathway, but just the difference between lectin pathway and classical pathway is the initiator of these two pathways. The initiator of classic pathway was the C1, but here MBL, mannose binding lectin, MBL. And MBL is a protein that can bind the sugars. For example, here, this lectin, MBL, can bind mannose. And there are two enzymes binded to MBL. They are called mannose binding lectin associated serine protease 1 and 2. And these two enzymes are similar to C1R and C1S. And this enzyme can cleave. These enzymes can cleave C4 and C2, and after cleavage of C4 into C4b and C4a, and after cleavage of C2 into C2a and C2b, C3 convertase form. As you see here, the C3 convertase is similar to C3 convertase of classical pathway. Then MBL is similar to C1Q with stalk and heads. And MBL can bind to mannose on the surface of microbes via of the head, through the head of MBL. And MAS1 and MAS2 are similar to C1R and C1S. And this C3 convertase can cleave C3 into C3A and C3B, and after cleavage of C3, the C5 convertase form, and as you see here, C5 convertase composed of C5b, C2a, and C3b, and this is similar to C5 convertase of classical pathway. And as you see here, different microbes can activate lectin pathway, bacteria such as Salmonella, Listeria, Neisseria, and fungi such as Cryptococcus neoformans and Candida albicans, and viruses such as HIV can activate lectin pathway. Okay, in this slide you can see the functions of complement system after activation. As you saw in previous slides, after activation of complement system, the cells can be lysed by the activation of some proteins of complement system on the surface of the target cells. As you see here, enveloped viruses such as herpes viruses, measles, mumps, influenza, and the gram-negative bacteria and other cells can lyse by activation of complement system. Another function of complement system is opsonization. During the activation of complement system, some fragments produce, and these fragments opsonize the bacteria and other cells, as you see here. So one of these fragments is C3b. C3b binds to immune complexes, and C3b has receptor on the surface of phagocytes, and as you see here, the immune complexes or bacteria can bind to the receptors on the surface of these phagocytes, and the phagocytes can phagocytosis the bacteria, and in this way phagocytosis facilitates. Other function of the complement system is increasing of inflammation. As you see here, complement system components involved in the extravasation. Extravasation means the migration of the inflammatory cells from the circulation into the inflamed tissues, and because inflamed cells such as neutrophils that involved in the inflammation has receptor on its surface, and some components such as C3a, C4a, and C5a can increase the extravasation and increase the inflammation. C3a, C4a, and C5a that produced during the activation of complement system, these are called anaphylatoxin, and these fragments can increase the inflammation by increasing the extravasation, and also that these fragments have receptors on the surface of inflammatory cells such as mast cells, as you see here, and induce the granulation of mast cells, and in this way can increase the inflammation. One of the most important of the function of the complement system is clearance of immune complexes. As you see here, after forming of these immune complexes in our body, they can deposit on the kidney granulum in the kidney, and on the endothelial cells of vessels, and glomeruli of kidney, and destroy the endothelial cells. That's why it is very important the immune complexes clear from the circulation. And after forming of the immune complexes, as you see here, the fragments of the complement system can bind to the immune complexes, and these fragments have receptors on the cells in the blood, such as phagocytes, monocytes, and neutrophils, and red blood cells, RBCs, and other leukocytes in the circulation, and these cells can carry the immune complexes in this way, carry the immune complexes into the spleen and liver, and in the liver and spleen, the immune complexes phagocytes and destroy, and in this way the circulation clear from the immune complexes. All of the function of biological function of immune system depend on binding of complement fragment to their receptors. As you see here, there are different receptors for complement components, and these receptors are found on different cells in our body. As you see here, CR1 is an important receptor for clearance of immune complexes. Then, as you see here, CR1 is found on the many cells: erythrocyte, neutrophils, monocyte, macrophages, and other cells. And three other important cells are important receptors for complement components fragments are C3A, C4A, and C5A and C5A receptors, as you see here, and they are found on the muscle basophils, eosinophils, granulocytes, and other granulocytes, and they are very important in the inducing of inflammation, because binding of these fragments to their receptors on the surface of these cells can induce the degranulation of cells, such as mast cells and basophils and other cells. Okay, in this slide you can see complement system activation at a glance, and after activation of complement system in three pathways, they can produce some fragments, and these fragments can increase the inflammation, can cause opsonization, and facilitates the phagocytosis, and also they can lysis, this fragment can lysis the target cell. Okay, deficiency of complement components or complement proteins can result in some problems and induce some disease, causes some disease. As you see here, homozygosis deficiency in C1q, C1r, C1s, C4 and C2 components of classical pathways results in immune complex diseases because of the position of immune complexes and deficiency in clearance of immune complexes, and induce diseases, autoimmune diseases such as systemic lupus erythematosus and vasculitis and glomerulonephritis. And also they deficiency in these components can increase the infections and recurrent infections by pyogenic bacteria such as streptococci and staphylococci. And also deficiency in C3 component. As you saw in previous slides, C3 has a central role in the classical pathway alternative pathway and lectin pathway, and the deficiency of C3 again, it can increase the immune complex diseases, increase the incidence of the immune complex diseases, and also increase the incidence of recurrent bacterial infections. And deficiency, homozygous deficiency in components involved in the MAC formation, for example, deficiency in C7, C6, and other proteins in MAC pathway can increase the recurrent meningococcal and gonococcal infections caused by Neisseria species, because these bacteria are very sensitive to MAC formation, and they can lyse by MAC formation. You can see here SLE patients with some manifestations, features of the disease, butterfly rash on the patient face, and hair loss, and deposition of immune complexes in the skin. This is an immunofluorescence test, and in this test you can see the deposition here of immune complexes on the epithelial cells of the skin. And these patients have different clinical manifestations, as you see here. And in this slide, what is important is the decrease of C3 and the C4 levels, serum levels, as you see here. For example, C3, the normal range of C3 is 90 to 180 milligram per deciliter, but the serum level of C3 of the SLE patients or this patient is 67. Then deficiency in the protection of complement components, especially C3 and C4, or components in the classical pathway, can increase the incidence of the SLE. And also MBL deficiency can result in the serious pyogenic infections in neonates and children. Okay. In this part, I want to talk about the regulation of the complement system. There are proteins in the complement system, and they can regulate the activation of the complement system, and or improper activation or a high activation of the complement system. I'll talk about the regulatory proteins in the complement system in this part. C1 inhibitor is a regulatory protein in the classical pathway, and it can dissociate the C1R and C1s from C1q in C1 molecule, and in this way can inactivate the C1 molecule, and after dissociation of C1R and C1s, the C4 and C2 cannot activate, and the production of C3 convertase inhibited by C1 inhibitor. This is a patient with C1 inhibitor deficiency. This disease is called hereditary angioedema, and these patients have a systemic edema, as you see here, edema around their eyes, eyelid, and edema around their lips. And this is in this slide you can see the regulators of classical and lectin pathways. Regulators can act just for classical and lectin pathways activation. C4b binding protein, C4b binding protein, and complement receptor 1 and membrane cofactor protein. These are three proteins can regulate classical and lectin pathways. As you see here, these three proteins bind to C4b and prevent the binding of C2 to C4b and then prevent the formation of C3 convertase. After this prevention, C4b cleaves by factor I into smaller fragments, C4d and C4c. C4c is soluble and C4d is a bound protein that bind to the surface of the target cell and can function as an opsonin. Factor H, CR1 and MCP again are regulators of alternative pathway. Factor H is a specific regulator of alternative pathway. And here also these three regulators bind to C3b in alternative pathway and prevent the formation of C3 convertase in alternative pathway. And again here C3b cleaves by factor I in two steps. First, IC3b produced, and after that, IC3b cleaves again by factor I into a smaller fragments, C3c. C3c is soluble, but C3dg and IC3b are bound cells, bound on the target cells. And C3f is also a soluble factor or fragment. Okay, I described a case at the start of the lesson, and you saw there that the patient has a deficiency in C3 and factor B, or low level of C3 and factor B in his serum. But after investigation of this patient's genetic investigation of this patient, it is found that this patient has deficiency in factor I, genetic deficiency in factor I, not genetic deficiency in factor in C3 or factor B. And now the question is how the deficiency of factor I results in the deficiency or decreased level of C3 and factor B. Please answer this question in Navid site and send me in Navid site. Okay, DAF is another regulator that function in three pathways: classical pathway, lectin pathway, and alternative pathway. DAF is abbreviation of decay accelerating factor. It is also called CD55. And the function of the DAF is preventing the function of C3 convertase in this way. DAF binds to C3 convertase in three pathways and dissociate, for example, in classical pathway, C4b from 2a, and in this way can inactivate the C3 convertase in the classical pathway, or in alternative pathway can dissociate C3b from bb and inactivate the C3 convertase in alternative pathway. As you see here, again, C4b and C3b after dissociation of C3 convertase or destroy of C3 convertase, factor I cleaves these two fragments into smaller fragments. S-protein is another regulatory protein in complement system, and this protein regulates assembly of MAC, regulate the MAC pathway. As you see here, C5b67 after formation can release from the surface of target cell and bind to the host cell and destroy the host cell or nearby cells. And S-protein bind the free C4b67 and prevents the binding of this free complex to the nearby cells, and this prevent the destroying the nearby cells in this way. Another regulator of the complement system that functions in the MAC pathway is homologous restriction factor, HRF, or CD59. And this regulatory factor binds to C5b678 and prevents the polymerization of C9 around the C5b678, and in this way prevent the again the nearby cells from the lysis, because C5b678 also can lyse nearby cells or host cells. That's why this regulatory factor bind this complex and prevent the polymerization of C9 around the this cell, this complex, and prevent the lysis of host cells. DAF and CD59 deficiency results in a disease called paroxysmal nocturnal hemoglobinuria, and this is a disease that because of not having that DAF and CD59 on the cells in our body, these cells are very sensitive to destroying and These two regulatory factors or regulatory proteins are found on the surface of all cells in our body. For example, they are found on the surface of RBCs, leukocytes, platelets, and other cells in our body, and these two proteins prevent the cells from destroying by complement system. That's why not having these proteins on the surface of the cells results in the destroying of cells such as RBCs. That's why these patients have hemoglobin or lysis of RBCs in their urine, in the urine of the in the morning. And the deficiency of the expression of CD55 and CD59 on the surface of the cell, it's not because of deficiency in the production of CD55 and CD59. For expression of the CD55 and 59 on the surface of the cells, these molecules need GPI for expression on the surface of the cells. And CD55 and CD59 anchor GPI in the bilayer membrane and express on the surface of the cells. And the deficiency of expression of CD59 and CD55 is not because of genetic deficiency of these two proteins. It is because genetic deficiency of GPI. GPI is abbreviation of glycosyl phosphatidylinositol, and deficiency in the expression, as you see here, of GPI results in the not having CD55 and CD59 on the surface of the cells in PNH disease. Okay, there are two questions in this quiz. Please answer these two questions and send me in Navid site, and nothing else. I wish success for my dear students. Good luck.